aim of the experiment is to be able to follow a population of dividing SIR1 cells under the microscope so that I can see how the epigenetic states change, how often the epigenetic states change with each successive division of the cells.
Take a long cover slip. Take about 7 microliters of cells. Place on the long cover slip and use a synthetic media plate. Take a razor blade and then make cuts into the agar. So take an agar block, lift it up, and place over the cells. Okay. For short-term experiments up to 3 hours, this by itself is fine. The cells will be immobilized. For longer-term experiments, I add liquid media to the top of the agar pad. And then put a glass slide over top of the whole thing. The media will provide us like a seal between the two. The slide is solely to prevent evaporation from the top of the agar block. So with this setup, the agar block will remain moist and the cells will continue dividing for at least up to 8 hours.